Okay, so this is lecture 19, and in the last class we finished our discussion of the mitochondria. And one of the ideas is that we have these two coupled processes, these linked processes, that actually function independently. The first is the electron transport chain, which produces a proton gradient. And in the second step, we utilize that proton gradient to make ATP. And the fact that these were coupled, but not part of a single process, was demonstrated by Racker and Stokenius. And they used these synthetic vesicles where they had ATP synthase from a cow. And they combined that with an enzyme or a protein called bacteriorhodopsin. And bacteriorhodopsin, in response to light, moves a proton across a membrane. And what they found was that in the dark there was no ATP production, but as soon as they turned the lights on, this would produce ATP. And so clearly from two different organisms, two different domains in fact, being that the bacteriorhodopsin is bacterial or archaeal, and bovine is, of course, eukaryotic. The way that ATP synthase works is that the proton gradient, which would be, in the case of the mitochondria, we have high concentration of protons in the intermembrane space, so it's favorable for them to move into the matrix. And in so doing, they turn this C component of ATP synthase, which turns the gamma stalk, and as the gamma stalk moves, sweeps through the beta subunits, it alters the structure of those subunits, therefore altering the function. And in one complete turn of ATP synthase, 3 ATP are produced. And it's important that these ATP are produced by taking ADP and adding an orthophosphate, which is an inorganic phosphate, onto the ADP, rather than transferring that phosphate group from another type of intermediate. It's actually the inorganic form that's used. And if we kind of do the math around this, even though it takes 10 protons to do a full turn of ATP synthase, we talked about how every proton that moves up the gradient via the electron transport chain, not every one of those flows through to produce ATP synthase. Some of those other protons are used to transport other molecules. So for approximately every 4 protons that move across the inner mitochondrial membrane, we produce 1 ATP. And we care about that because we know that if we are donating electrons from NADH, we move 4, 8, 10 protons for a total of 2.5 ATP. And for FADH2, we move 4, 6 protons for a total of 1.5 ATP. Okay? So another form of ATP production utilizes the same principle. A proton gradient is built, and from that proton gradient, we can utilize ATP synthase to drive ATP production. In this case, however, what's moving the protons isn't an electron transport chain, but instead is this photosynthetic system. And the reasons that these are a little bit different can be seen here. So when we're looking at the previous system where we were harvesting the electrons from sugars or fats, moving those electrons onto NADH or FADH2, those are very good electron donors to an electron transport chain. In contrast, when we're looking at plants, they get their electrons from water, and the electrons can't really flow from water. They can't flow from water onto NAD+ or FAD. So they can't flow directly in there. So what we do is, so we can't do much with those electrons. So what we do is we energize them with light, as can be seen here. And then from that, we can move them through basically a series of redox centers and move protons. And we can also produce NADPH, which will be a source of electrons when we go to build carbohydrates. So in this slide, we can see the things that we produce from the phototrophic part is we make that proton gradient and therefore make ATP. And we can also produce NADPH. And the third thing we need then to make carbohydrates or carbon dioxide, and that comes from the atmosphere. You can see that in a little bit more detail here with the two photosystems. Keep in mind that the one that we may think of as the second photosystem is actually photosystem 1. Photosystem 2 is used for harvesting the electrons. Photosystem 1, we can either produce NADPH, or we can keep cycling this. As long as those electrons stay in here, we can keep moving protons and keep producing ATP. And so the last thing we looked at was the Calvin cycle. And for this, 3 carbon dioxides come in, and we get 1 3-carbon sugar, glyceraldehyde 3-phosphate out of it. Okay. And that glyceraldehyde 3-phosphate can then be built into other carbohydrates. Okay. So that is where we were at before last week. Not all the students have written the exam yet, so I can't give you your exams back, but I know some students are worried because the drop date is Sunday, so you will get your grade, but that's it for the exam, and you won't get any feedback until next week. So the exam, you'll see just your grade. That'll be out of, again, 27 this time, because there was one kind of bummer question that we'll learn about next week. And there was a couple of bonuses. So it's you'll get your grade today or tomorrow. Yeah. Okay? And it was fine. I think the average was like low 70s, so good. That's what we were hoping for. Hey, that's enough excitement for one day. All right. Okay, so today we're going to be switching to our next topic. We're going to look at translocation into the ER, post-translational modification in the ER, and drug detoxification. And we may talk a little bit about trafficking through the Golgi. So this is topic 4, and it's going to build on some of the information we learned in the last section, building on ER, Golgi, and lysosomes. So again, these are the ebook sections, as well as the pages if you have the hard copy. So some people wanted the old 8th edition material. I can't verify the accuracy of this, but I've included what we used to use as the readings, although the course has shifted a little bit. So when we look at the endomembrane system, what we have is the endoplasmic reticulum, we have the Golgi, and we have lysosomes. We also have a whole bunch of different types of vesicles that we're going to discuss. And so the vesicles are how we move things around in this system. We move it between organelles, but this is also how we move things between, say, the plasma membrane and the organelles, or the plasma membrane and the cytosol. The textbook in this particular diagram has peroxisomes included, although peroxisomes are actually not a part of the endomembrane system. So the biggest organelle we have here is the endoplasmic reticulum. Endoplasm means within the cytoplasm, and reticulum just means membranes. And the amount or the surface area of membrane can account for over 50% of the total membrane in a cell. So it takes up a lot of surface area in the cell. And in cells that specialize in secreting proteins, I think we looked at the example of a pancreatic cell a couple weeks ago, they will have a very high percentage of their membrane devoted to endoplasmic reticulum because the ER is very important for the production and processing of secreted proteins. When we think of it, there's a few things we think of. First of all, production and modification of proteins that are bound for secretion or that are going to become membrane-bound proteins. But this is also the site where we have lipid biosynthesis, so most lipids are made on the surface of the endoplasmic reticulum, either the rough or the smooth ER. So we'll start with the rough ER. Remember, these can be separated, the two types of lipids, by, of ER, by homogenizing the ER, so basically breaking it up, and then it will form these little vesicles, okay? And through these little vesicles, we can separate them based on their density because the rough ER has ribosomes associated with the surface. And they're not covalently attached, the ribosomes. They're non-covalently associated with these complexes called translocons. And the translocons are used for bringing materials across the membrane. It's also the site of lipid synthesis. So just a note here, when we're talking about lipid synthesis, most of it happens in the ER, but it does happen on either production or modification happens in association with other organelles, especially the mitochondria. The lipids themselves are produced on the cytosolic phase of the ER. So let's just pretend it looks like this, and we have our two membranes. We're looking at like a cross section, and this is the lumen, so inside the ER. The production occurs on the cytosolic phase, and what that means is that that membrane would continue to get bigger and bigger and bigger. So we have to utilize transport proteins to flip those lipids through the membrane, because we can imagine if only the cytosolic space is having new lipids added, it's going to just keep expanding. And then it'll start kind of wrinkling up or whatever it would do. So there's a bunch of active carriers to move those lipids through. In terms of protein production, the proteins, of course, are made in the cytosol by the ribosomes. And the textbook goes to some lengths to talk about bound versus free ribosomes. And what they don't really focus on is that, well, I'll back up. So bound ribosomes, the proteins that are made here are co-translationally translocated, meaning that as they're being translated, they're directly being translocated. And I've got a couple slides. Well, we're going to do a whole section on that. But there's lots of other proteins made, and what they're kind of drawing the difference between here in the textbook is they're showing, in this left-hand panel, they're showing a co-translational translocation, co-translational translocation. And what that means is that as the protein is made, it's directly extruded or passed into the ER lumen. And on the right, we're seeing that there's lots of other proteins made by free ribosomes. And what they're demonstrating here is that once a protein is made and folded up in the cytosol, these are showing some of the different locations it's moving to. What this slide lacks to show is, in fact, some ribosomal proteins are made in the ribosome first, and then they get unfolded and translocated into the membrane in a separate step. And we're not going to focus on that mechanism of translocation. We're going to really be focusing on the one on the left here. So if we're trying to figure out how this works, so the way these experiments were done was that, and I'm just going to draw one of the membranes because we really don't need both. So here is a microsome, and it's from the rough ER, so it's got a whole bunch of ribosomes associated with it. And what you do is you provide amino acids, the 20 amino acids, and some of them are radio labeled. And that just means we can follow where they go. And if you take this and you incubate it with the other components required, so this is incubated with all of the components required for translation to occur in vitro, and we let it run for 1 or 2 minutes, and then you can stop translation and release. You can stop translation and release the protein from the ribosomes. And so there's 2 possibilities here. The first would be that proteins are made in the cytosol. All right, so proteins would be made in the cytosol. And when they're released then, you would expect them to be floating around out here. If, however, they're co-translationally made, we would expect them to be inside the microsomes. Okay, so those are our 2 possibilities here. So we run this experiment, we pellet out the microsomes. Okay, so you pellet out the microsomes. So they're down here. And here's our supernatant. So where does the radioactivity end up? Well, it turned out that the radioactivity ended up here in the microsomes. That's where the proteins were located. And so this was how they demonstrated. This was work done by Sabatini, and the second name escapes me at the moment. And so this demonstrated that as the protein is being made, it is directly passed into the ER lumen. So that was a little experiment that was done to show that. Because otherwise, if it was built in the cytosol and then imported, we would expect to find at least some radioactivity in the supernatant, and they didn't find that. I should point out that the radioactive free amino acids were filtered out of this, so they didn't show up in the supernatant fraction. Okay, questions about that? So kind of a neat little experiment that built on our knowledge of separating the microsomes. Okay. So there's a few slides with descriptive text here, and there's others. I've also included the diagrams of it. So I'm gonna work through the diagrams, but you can refer to the text for your kind of study purposes. So initially, we remember that proteins are made from N to C terminus. And we know that they're built inside this complex called the ribosome. And that ribosome can be seen in this slide as the green thing. And so as the protein is made, the N terminus is the first part to emerge out of the ribosome. And the N-terminus is where we find the ER localization sequence. And the localization sequence has 3 parts. Okay, so if we were looking at the N part of the terminal of the sequence, and then here would be sort of the C-terminal part of the sequence, and then this goes on to the rest of the protein down here. So here there is a bunch of positive charge residues right at the end. And then there's a bunch of non-polar, a little stretch of those, and then a stretch of polar. So there isn't a consensus sequence, meaning a specific sequence of amino acids that's recognized, but there's modifications based on this. And that's not hugely important for memorization purposes. As this thing emerges from the ribosome, it's bound by this complex called SRP for the signal recognition protein. And this protein is actually what we call a riboprotein. It's made up of 6 polypeptides and an RNA. And so there's actually a functional component of this is RNA. Okay? We talked about how proteins do almost all the stuff in the cell. In this case, we have an RNA component that's required for this to be active. When SRP binds to that signal, it actually stops translation. So the ribosome completely stops what it's doing. It recognizes, the SRP recognizes a receptor on the surface of the ER, and that receptor is part of the translocon, which is what is going to ultimately translocate that protein into the lumen. So this binds, we then get 2 GTPs coming in, okay? And both the receptor and the SRP bind to GTP, and both of them have GTPase activity. So those 2 GTPs get hydrolyzed, all right, to GDP, and this restarts translation. Now, as translation occurs, maybe I'll use a different color here. As translation occurs, the protein now we see is no longer, doesn't get exposed to the cytosol. The signal peptide, the signal portion, is cut off by a signal peptidase. Remember, a peptidase is something that cleaves a protein, a peptide. And now we get translocation into the ER. The ER is chock full of chaperones. It's got a very high number of chaperones. So very quickly, our peptide, our polypeptide gets folded. And remember, it's folding in an environment that's similar to the outside. So if this protein is going to be secreted, chucked out of the cell into the exterior space, it's folded in such a way that it will be functional outside of the cell. Okay? That was one of the cool things about the endoplasmic reticulum. So here's sort of a fun fact. We talked about our disulfide bonds, and I talked about how under reducing conditions, we break those disulfide bonds. Remember? So under reducing conditions, our disulfide bonds go back to being sulfhydryls. Those are the conditions in the cytosol. Inside the ER, we have oxidizing conditions. And so in the ER, we can get disulfide bond formation. And that turns out to be important for the stabilization of proteins that exist outside of the cell. Because outside of the cell, in the general environment, the conditions in the body are oxidizing, generally. So that's just one of the neat differences that we can see between those two compartments. So questions about that, much of it? Okay. So we've got the importance here of the signal recognition particle. Sorry, I was calling it the signal recognition protein. It's signal recognition particle because it's not strictly a protein. It has an RNA component. All right, so I said that it's also important for transmembrane proteins. And the way that works is that we have a stop sequence. So in this slide, we can see, oh yeah, so we start the translocation. But when it hits this stop sequence, the translocation stops. And that translocon itself opens up. And it releases that stop transfer directly into the membrane. And so this is a mechanism by which we can get those transmembrane regions actually into the membrane. So what kind of amino acids do you think we're going to find in the stop transfer sequence of our 4 classes? Yeah, those are going to be nonpolar because they're ultimately, as we can see in the panel on the right, embedded into the membrane. So in this case, we would have a single pass transmembrane protein that goes across one time. There are, however, also start transfer sequences. And a start transfer sequence, the way it works is it is in an internal sequence, and that internal sequence, what it does is it causes translocation to start. And so here you can see an example of one of these. And in this case, what it's doing is it's getting to translocate C-terminal to the sequence. Okay, so here's the C-terminal of our protein. The C-terminal portion gets transferred. However, there's also start transfers that point the other way, that get the N-terminal end to transfer first. And so if that was the case, where would the N-terminus in this protein be? If this little green thing was flipped, where would our N-terminus be? In the lumen or the cytosol, assuming that this is cytosol, which I can't even spell today. Where would our N-terminus be? Who says the N-terminus, if we reverse that signal, flipped it, would be in the cytosol? Who says it would be in the lumen? Right, so it would be in the lumen. So this would give us a way, in this example, the way this transmembrane protein is arranged is the N-terminus is in the cytosol, and the C-terminus is inside the ER lumen. We're going to see that that's important because when this thing goes to the plasma membrane, that orientation remains the same. So if this moves to the plasma membrane of the cell itself, the N-terminus will be inside the cell, and the C-terminus in that example will be where? Outside the cell, exactly. But we can arrange it the other way because some proteins need to function with the C-terminus inside the cell and the N-terminus outside. We can also combine these, and there can be multiple start transfer sequences, start and stop. And so by combining start and stop transfer sequences, we can build multi-pass membranes, ones that move through the membrane more than once. So for example, if in this example there was a stop transfer sequence here, remember when that moves into the translocon, it would be released, and our protein in that case, our final protein would look like this. okay? Because here would have been our start transfer, and this would be our stop. So by combining starts and stops, you can build a multi-pass protein. Okay? People clear on that? It's a little bit tricky, but not too bad. And so this is how all of these transmembrane proteins are made. So they get embedded in the membrane very early in their little lives, and they remain embedded in the membrane throughout the entire endomembrane trafficking process, which we're looking at today. There's another fun little enzyme associated with the translocon, and this is the oligosaccharyl transferase. The oligosaccharyl transferase scans that protein as it moves into the ER lumen. And it looks for this sequence, asparagine X, serine threonine. What that means is that there's an asparagine. X means it can be any amino acid. And usually when they say any amino acid, it usually excluding proline and glycine. Because remember, proline and glycine are weirdos. Proline is that rigid one where the side chain attaches back to the amino group, and glycine has no side chain and no chirality. So generally, X doesn't include those. And then Sur/Thur means it can be a serine or a threonine in that position. The asparagine, okay, so we remember that the side chain at the end of that, we have this, okay? And this group here, the amino group here, is what attaches to the sugar or carbohydrate and leaves a nitrogen behind, and that's why it's called an N linkage, because it's an N-glycosidic bond. We learned as well that that sugar, it's the whole block of 14 building blocks that's added. And so this is showing it. Dolichol is a lipid to which it's built on dolichol and it's attached to that. That's a component of the membrane of the ER lumen. We can see that that asparagine then becomes N-linked to this, which is N-acetylglucosamine. And it's going to be linked through, remember, the anomeric carbon of this, because we always link through the anomeric carbon. So the asparagine links through the anomeric carbon of the N-acetylglucosamine, and it has this whole group of sugars attached to it. And those sugars play an important role in the folding of the protein, which you'll learn about in third year. And what happens is that basically it will always be modified. So you won't find proteins that have all 14 carbohydrate components here. Some are removed, and other carbohydrates will be added at later stages. We can also get modification of those through such things as adding phosphate groups to them. So there's lots of different things happen to that. Questions about the rough ER? Okay. So the smooth ER, again, it's important for lipid construction, but also lipid metabolism, including production of what are called lipoprotein particles. So lipids, because they're hydrophobic in nature, we can't really move them around easily. And so the way we move them around between cells, for example, is we move them between cells in what are called lipoprotein particles. And so we associate those lipids with a protein, and the protein basically forms a shield around them so that inside we have those hydrophobic lipids, and on the outside we have hydrophilic or polar proteins, so it can move through the space. And so the ones that you're probably familiar with, perhaps, are HDL and LDL, lipoprotein particles. So HDL is the thing we call good cholesterol, and that's one way of moving lipids through your blood. And LDL is what we call bad cholesterol, and that's also used to move lipids through your blood. And they're both lipoprotein particles, and these are constructed on the surface of the smooth ER. It's important for our calcium storage, which we'll see plays a role in cell signaling. Okay. This is where vesicles that are going to deliver materials to the Golgi, this is where they form at the interfaces between the smooth and rough ER. And the next part we're going to talk about is sort of fun. This is where the cytochrome P450 enzymes are found. So cytochrome P450 enzymes, I think in humans we have about 50 of these that will act on different substrates. At the top, we can see what they do is they take R, so R would be some drug, and they oxidize it to ROH. Okay? And so we add this hydroxyl group to it. This then is going to increase the solubility of the drug, okay? And it's also a site for further modification, which we'll look at in the second step. So remember, most drugs, okay, when they are, most drugs are hydrophobic in nature because they can move more easily into the cells. Sometimes this step is actually required to activate drugs. So when some drugs are designed, some pharmaceuticals, they're designed so that without this step, they're actually not active. And why do you think that might be? What happens as soon as, almost immediately when a drug is introduced into, say, the bloodstream? Where does it go? It goes everywhere, but it goes one very important organ almost immediately. It goes to your liver. So they pass almost immediately through your liver, and your liver is very highly enriched in smooth endoplasmic reticulum. And what happens in the liver is we get this oxidation occurring. And so for a drug, if that shuts the drug off, its efficacy drops almost immediately. But if that turns the drug on, activates it, then, as you said, because the drug is about to go everywhere, goes through the liver, gets activated, and then gets disseminated throughout the body. So some drugs are designed with this in mind. This is the actual chemistry that occurs. If you're interested, I'm not going to ask you about this, but it's information that I think several of you, I'm sure, will find interesting. So here we're looking at a sample drug. We're looking at ibuprofen. And if we look closely here, what the P450s do here is now we have this hydroxyl group added. So this is the oxidation step. For many drugs, the next step is what's called conjugation. And so in all of these examples, this big pink molecule gets added, okay, onto the drug, generally onto this OH group that's been added. So we add this hydroxyl group, and then we add a big polar molecule to it. And this then allows the drug to be excreted, and it will go out through either the kidneys in the urine or to the intestine through the fecal material. Okay? So the conjugation helps to inactivate, but also makes them very soluble at this point. And so this is a classic mechanism for ditching drugs. here is an example where we're going to see this glucuronic acid is going to get added. Okay, this is the group that's going to be conjugated here. So on the top left, we have a barbiturate compound. So these are utilized as sleeping aids, or were. I'm not sure if they're still used. These were actually first produced by Emil Fischer with someone called Joseph von Mering. So our old friend Emil Fischer, who's the bane of everyone's existence in the class probably, the Fischer projection guy. He was also the first person to synthesize caffeine though, so he did do some good in his life. And so here what happens is in the first step, we get addition of this hydroxyl group, and in the second step, we're adding on either glucuronic acid or this sulfate group. Sorry, so this is also shown on that slide. And then these will be excreted, and we get rid of the drug. Okay? Keep in mind, when we're dealing with drugs, we want them to have some level of persistence, but generally we want the body to get rid of them. Okay, some drugs we want to last for a short time, some to have a longer half-life, but ultimately we want to get rid of these from the body. Here's a negative outcome. This one is the big danger, the unknown danger. And so here we have acetaminophen. And acetaminophen, it gets modified a little differently, and it forms this complex here, this NAPQI. Now, in a perfect world, it will be conjugated to that sulfhydryl group of glutathione. So the G is just representing glutathione, which is this compound here, okay? And glutathione has this reactive sulfhydryl, so similar to what we would, it's just a cysteine. So, and that can, of course, interact. That's an active group. And so in a perfect world, that sulfhydryl will attach here, and this will be excreted. Okay? So that's a perfect world. The problem is that if the liver lacks glutathione, we know that proteins also have a sulfhydryl group on their cysteines. And what happens is if there isn't enough glutathione around, this attaches to liver proteins. And what that does is that destroys your liver. Okay? And so where we see this happening, and what makes this so dangerous, is that your glutathione levels drop after like a big night of taking drugs or alcohol. You take Tylenol or an acetaminophen because you're worried you're going to have a headache the next day, and what actually happens is it leads to liver damage. And this is the largest cause of non-chronic disease cause of requiring liver transplants in the Western world. And it's actually a huge number of people have to get liver transplants because of acetaminophen toxicity. So it's people treat it because it's over the counter as if it's a very safe drug, but in fact, it's not a safe drug if it's not taken exactly as directed. It's probably a drug that if it wasn't commonly used, you would never be able to get this over the counter, or probably even through current levels of testing. So, an interesting story. So be careful. Other functions of the smooth ER. So we talked about how when glucose moves into the cell, it gets phosphorylated. Okay? And also, here's our glycogen. When we store glycogen, Remember, we're storing all those glucose molecules. When we go to use them, we produce this phosphorylated form called glucose 1-phosphate, which is converted to this glucose 6-phosphate and can then be used in the process of glycolysis. But if we are, say, the liver, what the liver does is the liver regulates blood sugar levels. And what that means is that the liver has to be able to get glucose out into the blood. So liver cells, about 10% of their wet weight is glycogen. So they're a really good place for storing glycogen. Muscle cells, it's about 2% of the wet mass. But the problem is we know that the transporters on the plasma membrane can't move the phosphorylated glucose. And all the other cells in the body don't have any way to remove that phosphoryl group, but the smooth ER on the liver has this enzyme called glucose 6-phosphatase. And so in the liver, this enzyme associated with the smooth ER removes the phosphoryl group, producing just glucose, which then can travel out into the blood. And once it's in the blood, that helps to regulate the content of sugar in the blood. Okay? So part of this complex glucose metabolism system, we'll touch on that more in 299 as well, but that has impact for things like diabetes, for example. And so I think we talked about this. I think we'll stop here and we'll pick it up with this in tomorrow's lecture.